Antibiotic resistance occurs when bacteria develop or acquire the ability to survive antibiotics that would normally kill them or stop their growth. But how does this happen? Bacteria can develop resistance through changes or mutations in their DNA. They can also acquire resistance genes from other bacteria through horizontal gene transfer, including transformation, transduction, and conjugation. Now, when an antibiotic attacks a susceptible bacterium, it reaches a specific cellular target and interferes with an essential bacterial process. This may stop bacterial growth or eventually cause the cell to die. However, resistant bacteria have several mechanisms that allow them to survive. The first major mechanism is enzymatic destruction or modification. Some bacteria produce enzymes such as beta-lactamases, which can break down or modify antibiotics like penicillins and cephalosporins, making them ineffective. The second mechanism is target alteration. Genetic changes can modify the structure of the antibiotic's target, so the drug can no longer bind properly. The bacterial process can then continue despite the presence of the antibiotic. The third mechanism is reduced permeability. Especially in gram-negative bacteria, changes in the outer membrane or membrane channels called porins can reduce the amount of antibiotic entering the cell. But even if the antibiotic enters, bacteria may use another defense called active efflux. Specialized proteins called efflux pumps actively remove antibiotics from the bacterial cell, lowering their concentration and reducing their effectiveness. Bacteria can also use target protection or metabolic bypass. They may produce proteins that protect the antibiotic's target or use an alternative pathway to continue an essential cellular process that the antibiotic has blocked. Resistance can also spread from one bacterium to another. For example, during conjugation, a resistance plasmid can be transferred from a resistant bacterium to another bacterium through a cell-to-cell connection. The recipient can then acquire and express new resistance genes. Now comes an important process called selection. When an antibiotic is introduced into a bacterial population, susceptible bacteria are eliminated, while resistant bacteria survive. These surviving bacteria multiply and pass their resistance traits to the next generation. With continued antibiotic selection pressure, resistant bacteria can become more common in the population. Over time, bacteria may acquire resistance to several different antibiotic classes. This is known as multidrug resistance, making infections much more difficult to treat. So, bacteria can resist antibiotics by destroying or modifying the drug, changing its target, reducing drug entry, pumping the antibiotic out, or protecting and bypassing its target. Resistance genes can also spread between bacteria.